stratification and tumor board. So Sam, I'm going to let you take it away.
Okay, so hi everyone. For those who don't know me, my name is Sammantha Avaylon, and today I will be presenting my thesis project, which was focused on graph-based multimodal integration for breast cancer subtype classification. And so to start off with, I just want to give a broad overview of breast cancer. And so in 2024, the SEER database reported breast cancer to be the most common type of cancer in the United States, attributing to 15.5% of all new cancer cases and 6.9% of all cancer-related deaths. These statistics can be partly attributed to the tumor's inter as well as intra-heterogeneity, which is caused by variation in the genomic, transcriptomic, proteomic, as well as epigenomic profiles of the tumor. And so subtyping is a critical tool to navigate heterogeneity through diagnostic tools such as histopathology, immunohistochemistry, as well as gene expression assays. Gene expression-based molecular subtyping specifically has significantly enhanced patient prognosis as well as the creation of targeted therapies. However, there are challenges associated with this method, which I will touch upon later. And so this really highlighted the clinical need for alternative approaches for molecular subtyping. And so first, I think it's important to highlight the complementary methods used alongside gene expression-based molecular subtyping, as there are various factors that affect patient prognosis, where tissue morphology is one of those key factors. And so to analyze tissue morphology, a tissue biopsy sample is first collected from the patient and stained using hemoxicillin and eosin. And so by staining this tissue, a pathologist is then able to analyze key morphological features that help them predict histological subtype as well as tumor grade. And so tumor grading specifically measures the tumor's aggressiveness by essentially measuring the degree of cellular differentiation. And so through this method, a pathologist is able to see how similar a tumor tissue looks compared to normal tissue. And so this is done on a scale of 1 to 3 and is based off of 3 key morphological features, including mitotic count, nuclear pleomorphism or atypia, as well as tubular formation. And so briefly, mitotic count reflects the rate of tumor cell division as well as proliferation, whereas nuclear pleomorphism measures the size and shape of the cancer cell nuclei, whereas for tubular formation, it measures the presence of tubular structures in the tissue. And so if we look at our image to the right, we could see how tubular formation actually decreases with increase of grade. And so this means that the presence of more tubular structures is more indicative of normal breast tissue. Whereas for mitotic count and nuclear pleomorphism, this does increase with tumor grade. And so while traditional methods like H&E analysis are essential for breast cancer diagnoses, the variability in treatment response within patients within the histological subtypes highlighted the need for further subtype refinement. And so advancements in gene expression profiling have significantly enhanced our understanding of how molecular heterogeneity drives breast cancer prognosis. And so in 2000, by using cDNA microarrays to study the gene expression of 78 breast cancer samples, through hierarchical clustering analysis, Prue et al. was able to identify the 5 molecular subtypes, including luminal A, luminal B, HER2 positive, basal, as well as normal-like. And so if we look at the table at the bottom left, we could see that these molecular subtypes can also be characterized by immunohistochemistry status as well as tumor grade. And so briefly, luminal A and luminal B are both within the luminal subtype. And so these two subtypes are characterized by having hormone receptor positive, so ER or estrogen receptor positive, as well as progesterone receptor positive, as well as HER2 negative. Luminal A and luminal B are generally associated with having a better outcome, where luminal A is associated with having the best outcome and is associated with a lower grade. And so if we look at the basal subtype, this is hormone receptor negative as well as HER2 negative, and this subtype is associated with a higher grade and therefore more aggressive tumor. And so research showed that gene expression-based molecular subtyping dramatically improved breast cancer prognoses, as well as the creation of more targeted therapies. And so this led to the creation of various diagnostic tools aimed at translating these molecular subtypes into the clinical setting, where specifically PAM50, or the prediction analysis of microarray 50, is considered to be the gold standard for molecular subtyping. And so how this is done, they first measure the expression levels of 50 genes that have been significantly linked to breast cancer prognosis, and we can see these 50 genes in our table at the bottom. And so by measuring the expression levels of these genes, we're able to predict the different molecular subtypes shown in blue. In addition to this, this tool also allows clinicians to predict the risk of reoccurrence score, or so otherwise known as the ROR score, which allows the clinician to predict whether or not a patient would benefit off of adjuvant therapy. And so this really adds another layer of prognostic value to PAM50. And so, as I just highlighted, while the PAM50 assay is a critical tool to determine patient prognosis, as well as guide treatment decisions, there are challenges associated with this subtyping method, including high costs, as well as causing potential delays in treatment initiation. And so, based on the concept that tissue is the product of underlying molecular interactions, using H&E slide images is a promising avenue to increase the accessibility of molecular subtyping. And so an advantage of using this data type is that H&E slides are considered standard of care during diagnoses, as well as the fact that these slides are commonly digitally recorded as whole slide images. In addition to this, with the increase of digital pathology, these slide images have also become increasingly available in large public databases, such as the Cancer Genome Atlas. And so, considering this, we really were motivated to see if we can use H&E slide images alone or in addition to other data types for molecular subtype classification. And so research has shown the advantages of using convolutional neural networks to extract key morphological features from these images for disease-related subtyping tasks. And so for our first aim, we wanted to implement an attention-based multiple instance learning model to accurately classify basal and luminal TCGA breast cancer patients, and so breast cancer patients from the Cancer Genome Atlas database. And so by accomplishing this aim, we are then able to see if we can extract clinically relevant morphological features for basal versus luminal subtype predictions. And so here we can just see an overview of our unimodal model classification workflow. And so we could see that this workflow is broken up into 2 key steps, including histopathology, whole slide image preprocessing, as well as the attention-based multiple instance learning model. And so just to start going through this workflow, we first downloaded 716 whole slide images from the Cancer Genome Atlas and broke up the sample into training and test sample sets, where the training sample set contained 119 basal patients as well as 448 luminal patients, whereas our test sample set consisted of 34 basal patients and 115 luminal patients. And so if we look at the bar graph to the left, we could see that there is a class imbalance between basal and luminal, where basal is the minority. We did take this into account while training our multiple instance learning model, which I will touch upon later. And so once we have our images, one of the most critical components of our workflow is to extract clinically relevant image features from our whole slide images. However, before doing this step, we first need to pre-process our whole slide images because there could be variation in image quality, which could arise from either differences in processing techniques, as well as the presence of unwanted slide artifacts. And so we can see examples of this in the bottom image where pen marks as well as tissue folds are unwanted slide artifacts that we want to avoid while extracting image features as they could negatively affect our model. In addition to this, given the large slot or size of our whole slide images, we need to divide these images into smaller patches to increase our model efficiency, as well as really be able to pinpoint which specific tissue regions we need to classify our basal and luminal subtypes. And so while doing this, a major challenge that arose was when the Cancer Genome Atlas does not provide slide annotations. And so annotations are normally done by a pathologist that essentially marks key tissue regions within the slide. However, without having these annotations, we were then faced with the challenge of how do we identify and extract patches containing these key tissue regions. And so to do this led us to benchmark several published tools, including Slidal, HistoQC, as well as SlideFlow, to segment the tissue regions from the background as well as from any unwanted slide artifacts. And so each of these tools leverage a combination of different thresholding techniques as well as deep learning models to create these tissue masks, as we could see in the first row, that essentially just segment our tissue region. And so to compare these 3 tools, we compared each of the tissue masks generated by each tool, as well as the quality of the patches extracted. And so if we look at our Slidal tissue mask, we can see it did a pretty good job segmenting the tissue, which is marked in blue, from the background, which is marked in green, as well as from the ink, which is marked in red. If we now look at our HistoQC tissue mask, which is marked in green, we could see that this tool severely undersampled our tissue region. And so this could negatively affect our model, as it might miss key morphological information that is needed to differentiate our basal and luminal subtype. And lastly, if we look at SlideFlow, where its tissue mask is marked in black, we could see that it oversampled the tissue region by extracting patches from the ink area. And again, this could negatively affect our model by introducing noise in our model. And so lastly, if we look at the bottom row, comparing each patch image, we can see that each patch essentially is the same from each tool. And so for our project, we decided to use Slidle as our pre-processing tool to not only segment key tissue regions, but also for patch generation. And so now that we have our patches, we then apply different transformation techniques to each patch, depending on if that patch came from either a whole slide image that was used for training or a whole slide image used for testing. And so for the patches that were used for training, we augmented these patches by using random flips, Gaussian blur, as well as color jitter. And essentially this just introduces variability within our sample set, which therefore improves the model's generalizability. For a test set, we just did standard color normalization because we don't need to introduce variability within our test set. And so after this, finally, to extract our clinically relevant morphological features from these H&E image patches, we used ResNet 18, which is a common convolutional neural network used to extract image features. And so a key characteristic of ResNet 18's architecture is its ability to capture global features such as edges and shapes in its earlier network layers, as well as more intricate and complex features and patterns from the network's deeper layers. And so by applying ResNet 18 on our histopathology data sets, we were then able to extract complex morphological features as well as tissue patterns as feature vectors that offer an overall comprehensive representation of each patch's tissue morphology. And so as we considered which supervised classification model to use for our project, one of the key aspects we had to consider was that not all regions of the tissue are actually informative of basal versus luminal classification. And so one of the key features of an attention-based multiple instance learning model lies in its ability to assign different levels of importance to each patch image through an attention mechanism. And so if we look at the bottom image, we could see that once we extracted our patch features using ResNet 18, we then fed these features through the attention mechanism. And so what this mechanism does is essentially just weights each patch according to its influence on the final prediction. And so by assigning higher weights to these key patches, the model can then create a single and comprehensive whole slide embedding, which is shown in blue in the bottom image, that summarizes and highlights the most important morphological features identified through the model. And so overall, what this attention-based multiple instance learning model allows us to do is to really capture a holistic view of the tissue while emphasizing these relevant regions for basal versus luminal classification. And then therefore this does improve model accuracy as well as the model's interpretability. And so this capability was reflected in our model's results, where we can see how during training it did achieve an accuracy of 82% and an accuracy of 79% for our test sample set. The macro F1 scores for both training and testing were the same as their accuracy results, showing that despite our class imbalance, the model still performed relatively well. Finally, to measure the model's ability to discriminate between basal and luminal subtypes, we used AUC. And so if we look at our graphs at the top, we can see that training achieved an AUC of 88%, whereas for our testing, we did achieve an AUC of 77%. And so this overall showed that our unimodal model did perform relatively well using just H&E slide images. And so the results from our unimodal model demonstrate that H&E images contain rich morphological information that can be used to differentiate between basal and luminal molecular subtypes. However, these results also show us that we can't use imaging alone to replace gene expression-based molecular subtyping, as it doesn't fully capture the molecular heterogeneity of these subtypes as RNA sequencing provides. And so with this led us to explore multimodal AI methods to see if we can accurately classify these basal and luminal subtypes. And so for our second aim, we wanted to implement a graph-based multimodal model to integrate H&E images and bulk RNA sequencing to accurately classify our TCGA basal and luminal breast cancer patients. And so through this approach, we really wanted to see if we can characterize as well as leverage the complementary relationships between our H&E images as well as RNA to identify novel genes outside of the PAM50 classifier and create a smaller gene set that could be used in addition to imaging for automated molecular subtyping. And so here we can see both our workflows for the unimodal model shown on top, as well as our multimodal model, which is shown in bottom. Before diving into the multimodal model classification workflow, I think a key aspect to highlight are the whole slide image features. And so as I previously talked about, we created these whole slide image embeddings in our unimodal model, which offers a summary and really highlights the key relevant tissue regions, basal and luminal classification. And so to increase the accuracy as well as interpretability for our multimodal model, we extracted these whole slide image, whole slide image features as our, we extracted our whole slide image embedding to serve as our whole slide image features for our multimodal model. In addition to this, for our RNA sequencing input data, we didn't just limit the gene set to RNA sequencing, or just the gene set to the PAM50 classifier. We used all the genes offered in the TCGA data set because we really do want to identify novel genes outside of the PAM50 that complement well with imaging for subtype classification. And so to dive into our multimodal model, we first downloaded the bulk RNA sequencing data available in the TCGA database for the H&E patients we used during our unimodal model. And this resulted in a total sample size of 716 patients. Again, we separated the patients according to whether they were for training or testing for our unimodal model. And so this resulted in our training set consisting of 119 basal patients and 448 luminal patients. Whereas for our test sample size, there were 34 basal patients and 115 luminal patients. And so again, there is a severe class imbalance, and this was taken into account for our multimodal model by incorporating class weights while training our model. And so to ensure data quality in our bulk RNA sequencing data set, we pre-process the data by first detecting and eliminating any potential batch effects, as well as filtering our RNA sequencing features to just the top 500 most variable features. And so now that we have both our data types, this then leads us to the question of how can we integrate these two data types. And so patient similarity networks offer a promising and interpretable tool for synthesizing different data types for both molecular subtype classification as well as feature refinement. And so patients who share similar clinical outcomes, as well as patients within the same subtype, often share similar clinical, molecular, and morphological profiles. And so we can represent these similarities as patient similarity networks, where if you look at our image, we can see that nodes represent patients and the edges between them represent their pairwise similarity. However, shown in our unimodal model, we saw that a single data type alone normally isn't enough to differentiate between patient subtypes. We can also see this in our image for A, B, and C, that we can't clearly differentiate the patient subtypes seen in the patient similarity matrices. And so to address this challenge, one of the key advantages of using patient similarity networks is that we can integrate these different data types and capture both the shared and complementary information from the different data types. And so if you look at our image, we can appreciate how the integrated network, as well as the Venn diagram, highlights how each data type contributes to the observed similarities between patients. And also, if we look at our integrated patient similarity network, we can also see that we now can differentiate the 3 patient subtypes. And so by leveraging this network, as well as the integrated patient similarity matrix, we can then use the shared global patterns among patients across different omic layers for subtype classification. However, a limitation with just using patient similarity networks is that it focuses on the global patterns shared at the population level. And so this just focuses on the interheterogeneity of disease. And so this overlooks intraheterogeneity, which is also a key factor in determining patient prognosis. And so for our graph-based approach, we decided to adapt the framework created by Duro et al., who used both individual networks as well as patient similarity networks for multi-omic integration, as well as subtype classification. And so if we look at our image, individual networks the individual networks, the nodes represent each feature. And so for our whole slide images, each node represents a whole slide image embedding feature, whereas for our RNA sequencing, each node represents a gene. The edges between these nodes are calculated using a node product algorithm, which is an algorithm that essentially measures how the expression of one feature correlates with the expression of another feature. And so once we have these individual networks, we are then able to create patient similarity networks using edge difference distance, where we can see now that each node represents a patient, and the edges between these nodes represent the similarity between different individual networks. And so by using both of these networks, we now are able to take into account both intraheterogeneity as well as inter-tumor heterogeneity to classify our basal and luminal subtypes. And finally, to integrate these different different data types, we use similarity network fusion, which is a graph-based algorithm that essentially fuses the 2 different patient similarity networks by focusing on patients who share a high similarity in one or more data types, while focusing less on patients who show a lower similarity. And so again, if we look at our image, we can see that the fused final network captures both the shared and complementary information between our whole slide images as well as our RNA sequencing. And so this approach really allows us to leverage the complementary relationships between these 2 data types for more accurate subtype classification. In addition to this, we are also then able to uncover any unique morphological or RNA features that may have been hidden when considering these data types individually. And so to classify our basal and luminal patients, we used a random forest classifier. And so essentially by using this approach, we were then able to use the network's affinity matrix, which is the integrated patient similarity matrix shown earlier. And by using this as input for our classification model, we are able to classify patients with unknown subtypes based off their similarity to patients with known subtype. And so to evaluate the overall model's performance, we used accuracy, macro F1 score, as well as AUC, similar to our unimodal model. And so seen here, we can see that our multimodal model results demonstrated superior performance compared to the attention-based multiple instance learning model, which used H&E images alone. And so this can clearly be seen where our training set achieved an accuracy of 97%, as well as a macro F1 score of 95%. Our test sample set achieved an accuracy of 96%, as well as a macro F1 score of 94%. Again, if we look at our graphs above, we can see that the training achieved, both training and test achieved 99% AUC. And so what these results really show us is that our multimodal model was able to characterize and leverage the relationship between RNA and H&E image features, as well as shows us the advantage of using this approach for molecular subtype classification. And so these promising results then led us to really dive deeper to see what specific RNA features or H&E image features are contributing to this model's successful performance. And so to do this, we use normalized mutual information ranking, or NMI ranking, which essentially measures how each feature influences or contributes to the model's results. And so by using this approach, we really are able to answer our initial question of can we identify novel genes outside of the PAM50 classifier, as well as identify a minimal gene set that could be used in addition to imaging for molecular subtyping. And so seen here, we could see our top ranked multimodal features, where the features marked in blue are our whole slide image embedding features, whereas the features marked in red are the RNA features or genes. Interestingly, the top 3 features are whole slide embedding features. And so again, these embeddings just provide a overall high level representation of morphological features from our whole slide images that were important for basal and luminal classification. And so while basal and luminal subtypes are defined based off gene expression, what these results really show us is that is the importance of imaging features in our multimodal model approach for molecular subtyping. Now focusing on our genes, on the table to the right, we can see our top ranked RNA features. And so seen here, 3 genes did overlap with our PAM50 gene set, including FOXA1, MLPH, as well as ESR1. However, we were able to identify 12 genes that are novel to PAM50, seen in our graph. And so to validate if these top ranked genetic features are truly informative of basal and luminal classification, we filtered our initial RNA input to just these 15 genes, as well as filtered our imaging data input to just the top 8 ranked features and reran our multimodal model. And so if we look at our results, just using our top 15 gene set plus our top ranked H&E image features, we can see that this filtered multimodal model did better than our initial multimodal model, where for training, we achieved an accuracy and macro F1 score of 98%, and where for testing, we achieved an accuracy and macro F1 score of 99%. Again, if we look at our graphs above, we can see that training and test achieved an AUC of 99%. And so what these results really shows us is that we were able to identify a reduced imaging and RNA feature set that are most informative of basal and luminal classification. And so to determine if our 15 gene set aligns with established biological distinctions between luminal and basal subtypes, we conducted reactome pathway enrichment analysis of our PAM50 genes, so the 50 genes I showed at the beginning, as well as our 15 gene set. And so if we look at our table, we can see that novel genes, TFF3 as well as AR, were enriched in PAM50 pathways. And so this included estrogen-dependent gene expression, simulation of intracellular receptors, as well as ESR-mediated signaling. Interestingly, 2 of our 3 pathways were estrogen-related. In addition to TFF3 as well as AR, several other novel genes that weren't enriched in PAM50 pathways have been significantly linked to ER expression and luminal versus basal differentiation in recent research. And notably, AGR2, AGR3, and Scooby2 were found to be positively correlated with ESR1, which is the gene that encodes estrogen receptor alpha. And so these findings align with established molecular characteristics that differentiate luminal and basal subtypes, where luminal is characterized as being estrogen receptor positive, where our basal subtype is associated or is estrogen receptor negative. In addition to this, research has also shown ER status to be significantly associated with specific histopathological characteristics, including tumor grade, tumor necrosis, as well as tumor elastosis, which is just the presence of elastic fibers in the tumor tissue. And so, again, tumor grade is measured by 3 key morphological features, where luminal A and luminal B have a lower grade, therefore better prognosis, whereas basal tends to have a higher grade and worse prognosis. And so for a luminal subtype, this means that they have more tubular structures present, as well as decreased nuclear pleomorphism and decreased number of mitosis, whereas for basal, because it is grade 3, this leads to a decreased tubular formation, as well as increased nuclear pleomorphism and mitosis. And so overall, the fact that our 15 gene set aligns with these established biological distinctions really highlight our multimodal model's ability to identify a gene set that accounts for the complementary relationship between morphological and genetic features. And so, encouraged by the results we saw from our 15 gene set, we wanted to really test the limits of our multimodal model and just use our top 3 imaging features and top 3 RNA features. And so this included FOXA1, AGR3, and TFF3. And so here we can see that our multimodal model that used the top 3 ranked genes as well as the top 3 image embeddings, this model did pretty well, where for training, we achieved an accuracy of 99%, as well as a macro F1 score of 98%, whereas for testing, we achieved an accuracy of 97% and a macro F1 score of 96%. And then looking at our graphs, we can see that both training and testing achieved an AUC of 99%. And so overall, what these results highlight is that the model is able to not only identify and select complementary features that can accurately classify basal and luminal subtypes, but also its ability to select a very small gene set that can be used in addition to imaging to address the challenges associated with molecular subtyping. And so while our approach demonstrated promising results, we did face a few limitations. And so first, in terms of our unimodal model, the image feature extraction could be further optimized to increase computational efficiency. In addition to this, as I highlighted earlier, during the attention-based multiple instance learning model, we were able to create these attention scores that weight different patches according to their relevance for basal and luminal classification to really increase the unimodal model's interpretability. What we could do is use these attention scores and essentially create these attention heat maps that could be overlaid on top of our whole slide image. And so that would really allow us to really visually see what specific tissue regions led to our results. And so for our multimodal model, we could improve the model by exploring other model parallel processing techniques to enhance the model's efficiency. Lastly, because our models were trained just using TCGA data to really see and test the model's generalizability, we do need to test the model on data sets outside of the TCGA database. And so to summarize my thesis project, again, current gene expression-based molecular subtyping methods, specifically PAM50, does face limitations in the clinical setting due to, especially due to delays in treatment initiation. And so our research aimed to address the challenges by seeing if we could use deep learning models to extract key morphological features from H&E images alone or in addition to gene expression for basal and luminal subtyping. And so although our unimodal model results showed great classification performance, they also showed us that we can't use H&E imaging alone for molecular subtyping. However, by using a graph-based multimodal model approach, we were able to characterize and leverage the complementary relationship between H&E image features as well as gene expression to not only identify a minimal gene set that could be used in addition to imaging, but also for accurate molecular subtype classification. And so overall, this suggests that our multimodal model approach is a promising method to address the challenges associated with PAM50, therefore improving precision oncology. And so for future work, we do want to run the graph-based model just using RNA sequencing alone to compare with the multimodal model, as well as see if we can just use the minimal gene set, either the 15 gene set or 3 gene set for molecular subtyping. As mentioned earlier, by using the multiple instance learning attention scores, we can create attention heat maps that highlight tissue regions most informative of basal and luminal classification. And so by comparing our identified regions to pathologist annotated slide images, we can see how our regions compare to established prognostically significant tissue morphology. And lastly, for dissemination plans, we do plan to create a code repository for our model through GitHub, as well as submit for a publication. Okay, so for my acknowledgements, I would like to thank the DMICE staff, Andrea and Vanessa, and Lynn and Lauren, as well as my TAC committee, my mentor Shannon, for your mentorship and guidance, as well as Mike, Guanming, and Andrew for also your guidance and support. And lastly, my friends and family. And that's it.
Great job. So we have time for questions for Sam. There's clapping. I don't know if you see the little virtual clapping. Or virtual clapping. Any questions for Sam?
I do have one question. Shannon, I think this is pretty amazing work. And I was trying to rack my head and answer the question myself, like what other places would this be very useful for? But I couldn't come up with any. But what do you think? I'm sure you've thought about this.
Well, other places. You mean what other data types we could use, or just?
Yeah, what other places would this multimodal graph-based method be used for? What are use cases?
Well, it definitely could apply to other cancer types, beyond breast cancer. In addition, we could use other data types as well to see, like we identified the, correlation between H&E imaging and RNA, but could we also identify any complementary relationships, for example, with proteomics or any other data type? Yeah, those are the first thoughts that come to my mind.
So Sam, maybe when you think about his question, you could think about just the multimodal approach in general, right? And the idea of especially the richness in the imaging, right? The H&E imaging, which is something we have a lot of, and we tend to think more, but we get more excited about the RNA-seq and these other aspects, right? So I think there's both the multimodal part and then also just the applications of really rethinking some of the common data types that we have clinically that contain a lot of information and may be helpful surrogates for, let's say more exciting technology, which is also more costly and more, less efficient in terms of time. I think it's both, right? Just to think about in terms of your bigger contribution.
Mm-hmm.
Great question.
Yes, Ryan.
This is awesome work. I guess my question is kind of using this as a discovery mechanism, right? I really appreciated your, I guess, feature collapse, and when you identified sort of the top ranked models, and sort of pairing off of what Amrish is asking, right? If you can include more modes of data, can you do the same process again, I wonder, and find other sort of high value top ranked features that may inform us on other ways we can make these diagnoses outside of RNA and imaging? And that's kind of what I'm wondering about.
Yes, we can definitely include other data types. One of the benefits of using the patient similarity networks is that it doesn't limit our ability to expand to other data types. And so, yes, is my answer to that question.
Yeah, I wonder how much value you could get from adding the radiology images of the actual patient's tumors. Obviously, it doesn't beat having the histopath of it, but maybe there's some features in there that could help.
Yeah,
Yeah.
definitely.
In the
Oh.
same line, like I wonder about time series data, right? Like in standard sort of clinical encounters where folks are kind of getting worked up in the outpatient setting and getting just standard basic metabolic panels, right? Or changes in there, right? May not be representative, but something along that line where you can extract a lot of information from serial clinical data. I think that'd be really cool too.
Yeah, those are definitely interesting future applications.
Any other questions for Samantha?
Hi, Sam. Great job. Just real quick question. I wonder, is there any data available that can kind of guide you in answering like how good does your classifier have to be? Like, you showed a really good performance with just 3 genes and 3 imaging features. But to have like real clinical benefit, does it have to be even reduced more than that? Is that good enough? I wonder if you have a sense of that.
Do you mean in terms of performance, like how well it has to be?
I'm
Or
thinking more of like a cost benefit analysis. You talk about the efficiency and that there's disadvantages to how long it takes and the cost of doing the full panel.
Mhm. That is a good question. I would have to think about that more.
So Sam, we've talked about this a little bit though, so I want you to think about Mike's question, right? So he's asking a question you haven't done, which is to benchmark in a way, right? You could say, we have the H&E, if you have the 3 genes, how long would that take? But there's also the question of how you do it. So what would be if you get down to 3 genes, we're not talking about RNA-seq anymore, right? What would we be talking about?
We could use like PCR.
Right. So then the timing issue, so then generating the data, H&Es are already there.
Right.
Time would be really fast. The real question, the last piece, which you have in benchmark, which would be really kind of cool, but is kind of beyond the scope of this, would be how long does it take us once the model's in place, then analyze the data and do it, right? You just want to make sure that that wouldn't be, but otherwise the data itself, the quantitative real time and the H&E, much more advantageous than RNA-seq. But you have to remember, because of the modeling piece, then you have to just ask how long would it take to get the answer.
Right. Right.
All right, any other questions before we let her go to her committee for the rest of her exam? Okay, if not, please join me in, again, congratulating Sam on a really beautiful presentation. And then for her committee, just a reminder, please, you'll come off this link and there's another link that we're going to join. Thank you all. Really appreciate you attending.
Thank you.